This is, if you go to school, there's always a point in the lesson where you have to take a test. So this is the test. Do you remember when Derek was talking about that field that was split into, and one was managed conventionally, anhydrous, a regular fertility program, and the other one was biologic? Remember that? I got the chance to visit that field twice. The first time with Ken, and then the second time, Dean and I were there. And the second time, we walked from the biologically managed field into the conventionally managed field. And if you remember, Derek was talking about the fact that the first thing that we noticed was weed pressure. The water hemp was just very, very thick on the outside edge, thinking, well, it's going to get better as we go. And it lessened, but it didn't get tremendously better. And we walked around in there a lot. We didn't what I would consider scout it. We just sort of made a foray in. And as we were coming out, I started noticing multiple pathogen diseases that were present on that conventionally managed corn. I found all of a sudden common rust. And then the next thing you saw, we found some southern rust. And it was just amazing to me, and really, if you will, flipped a light switch for me about the link between good nutrition, particularly late season nutrition, plant health, and then pathogen resistance. And so that's what we're going to talk about just a little bit. I wanted to put these numbers up here because I found this incredibly interesting. There was a study that was done recently that looked at the cost of disease on crops all across the world. And I used some very conservative numbers. But if you look at this in a 200 bushel corn crop, if you're losing 15% of that crop, now here's the thing. If I go out there and cut down 15% of the field, you're going to notice that. You're going to pay attention to that. However, if you never see it, in other words, the pathogen takes it and you've never noticed it, are you ever going to feel it in your wallet or your checkbook or anywhere else? And that's really what these numbers are talking about, that unseen cost of those things. And again, these numbers just assume a 15% loss. So $180 in 200 bushel corn, $85 in 65 bushel beans, and $47 in 60 bushel wheat. That's a lot of dollars that we could be leaving on the table. How much fertility, biology, or whatever would that pay for if we're not losing those? Has anyone ever seen this diagram before? Okay, see a few hands going up. This is what is called, sometimes you'll hear it referred to as the disease triangle. This is actually a pyramid, so it's just like the Egyptian pyramids. It's got three sides, but there's also the apex. And essentially what this diagram is telling us is, in order for us to feel the effects of disease, we have to have a pathogen present with a host plant in the environment all at the same time. And I like to use this example. Right now, currently, in every single one of your fields, you have pathogens. But you're not feeling you're not seeing the result of them because right now you don't have a host plant growing, more than likely, and our environment, we're too cool, we're too dry, whatever the case may be. However, in the summertime, when those things change and they all come together, that's when we're going to see these issues. There are a variety of ways that we might use to reduce our pathogen load or resistance. The one that I like to point out is over here, intercropping. And I like to tell this anecdotal story. My daughter got married this past summer, and she married a gentleman from Ireland. And so we've had the opportunity to go to Ireland a couple of times. And in Ireland, they do a lot small acreages. They don't measure things in acres, it's in hectares, but primarily a 20-acre field is a big field over there. And so there's a lot of intercropping that is done. So you might plant 4 or 5 rows of corn, 4 or 5 rows of beans, 4 or 5 rows of another crop. There's a lot of that that's done. Really, small cereal grains are about the only thing that's planted in a homogenized way. The fascinating thing, you can reduce disease by 75% utilizing that intercropping method. Now, does that mean that's the best way to go? Not necessarily, but it is interesting. Obviously, we've been talking a lot about inoculation, where we get a direct and indirect antagonism of that. And we're gonna talk about what that means here in just a minute. What we really want to focus on is what Ken was talking about, that crop nutrition. We talked about early on how we set ourselves up for success, and then how throughout the growing season, we continue to monitor that crop through the use of tissue test and sap analysis and all of that to make sure that we are maintaining an adequate and balanced nutrition within the plant so that it can protect itself. We talked about the Mulder's chart this morning and how we sort of use that, but I also wanted to show this. Essentially, when pathogens attack plants, what are they looking for? Weak spots. Oftentimes, what you find is a disease like sudden death syndrome. You have some type of a soybean cyst nematode that will do some stinging under the ground at the root level, and the pathogen will enter there. And then the next thing, not the next thing, it might be 2 months later, you begin to notice the disease, even though that infection started very early on. And the same is true of above ground pathogens. They are looking for sites where they can get in. And healthy plants tend to have the ability to lower that susceptibility. They can keep their stomata closed in a more consistent fashion, opening only when they need to. They also have a thicker cuticle on the leaf surface. So when a fungal body lands, it's not nearly as able to get through that cuticle layer and put its roots down, if you will. Fungal bodies actually have a quote unquote root system in order to get into the plant. And then, of course, we have all of these other ways in which this is happening. Proper nutrition helps the plant in a direct fashion through what we call redox enzymes. Essentially, pathogens are oxygen-loving organisms, and we have these things within plants called redox. They reduce oxygen in such a way that it limits the oxygen so that the pathogen can't actually grab onto that oxygen and use it to grow, to propagate. Now, it's not going to change the infection site. What it changes is its ability to continue to grow on that plant. And then in an indirect way, Ken kept talking about root exudates and how what the roots are putting out, it's feeding the plant. By altering those root exudates, we're able to grow good bacteria below the soil that are eating up the nutrients. Guess what else needs those nutrients? The pathogens need those nutrients. And if they're not there because the microbes are chewing on them or the plant is chewing on them, it's able to eliminate some of those things. And then if we just look at the sort of the three big ones, nitrogen, phosphorus, potassium, we know that when we have excessive nitrogen in the plant leaf tissue itself, that cuticle begins to get thinned out, particularly high salt nitrogens will thin that out. But when we just have high nitrogen, it limits that cuticle development. And so we have a higher susceptibility of disease. Phosphorus, we've talked a lot about phosphorus and how we don't pick it up in cool soils. The majority of our seedling diseases affect plants very early on. And phosphorus is very good at promoting root health and root development. And so we have these pathogens entering the plant through the root. When we can't pick up phosphorus, we have vulnerable roots. And then finally, potassium, the nutrient that's becoming my favorite. We have these positively charged potassium ions in the xylem and the phloem, as we were talking about this morning. And they have the ability to sequester disease. Disease, once it enters the plant, it will also enter the xylem and the phloem and move throughout the plant. And potassium has the ability to sequester that and keep it from moving around within the plant. When we have all of these things working in balance, it sets in motion what we call a cascade response, where one thing lessens the pathogen ability to implant itself within the leaf. The next one lessens the ability of the pathogen to grow in the leaf. The next one lessens the pathogen's ability to spread within the plant. So we get this sort of cascading effect. And then the other one, manganese, is a very key nutrient. In almost, if I were to poll the seed guys in here, almost all of them could tell me about their corn hybrids and soybean products that maybe are really good at limiting a disease, like say Phytophthora or something like that, because they have these little triggers within them that turn on and turn off based on the presence of certain nutrients, and then they're able to fight off these particular nutrients or pathogens. And manganese is one of those nutrients that is key to turning the light switch on and turning those triggers on, particularly like the RSPC1 Phytophthora gene in most soybeans. It's very key. Okay, here comes the test. Are you ready? You've been sitting, listening. Now's your opportunity to talk. I'm going to show you some pictures of some different diseases, and let's see if you know some of these things that we are looking at. Anybody ever seen some soybean plants in the field that looks like that? Anybody have an idea what we might be looking at? Let me give you a couple of hints. Very good. This is a resident disease that is present within the soil. It favors cool, wet soils, generally poorly drained soils. It is one of those things that you don't see the infection, you see the result. All of a sudden you're walking through your field and you see a clump of dead plants. Now that clump can be anywhere from the size of Gary's computer to the size of Gary's car to the size of Gary's house. I don't know how big that is. To the size of Gary's street. It can just grow depending on how bad the waterlogged place is. That is in fact Phytophthora. Most soybeans will, I shouldn't say most, many of the soybeans sold in the industry will tell you they have resistance to Phytophthora, which is true. What they don't tell you is they have a single gene that will generally suppress 1, 2, or 3 races of Phytophthora. Anybody want to venture a guess how many different races of Phytophthora exist in U.S. soils? Higher. Not that many. 21. There are 21 different races, and the majority of our genetics only are effective on certain ones. A telltale sign of this disease is this reddish growing lesion that will grow up the stem. And I say reddish because it really, sometimes you get brown lesions. This one is really red. It gives you that sort of reddish appearance. Okay, how about some others? Any idea of what this disease is that we're looking at? Brown stem rot. How many of you think it's brown stem rot? Okay, we got some brave souls. How many of you think it's not brown stem rot? It is not brown stem rot. There are diseases that are reflected here, but we classify these as seedling diseases. They affect soybeans. And so you have these different seedling diseases. Rhizoctonia here, even though it is favored by cool, wet soils, it shows up in dry conditions, typically drouthy type soils, sandy soils, heavy clay soils. Pythium will tend to show up in your wetter, poor draining soils. And then you have Fusarium down here in the lower left. A couple of key things about them. Obviously, on Rhizoctonia, you get these lesions that form at the base of the stem. Pythium, you notice sometimes this is called dampening off or damping off because you can see the taproot is really beginning to just dry up and sort of turn into nothing. And then the fusarium disease, you can see what's happening. It's infecting the vascular tissue, the xylem and the phloem spreading outward, limiting nutrient and water uptake. Okay, any ideas what we're looking at here? It's tar spot. Again, this is a resident disease, tends to favor cooler, wetter soils, likes extended leaf wetness, long periods of time. We had never seen tar spot in Southwest Missouri. I noticed tar spot in Indiana in 2015, I believe. Hadn't seen it this year, was the first time that I saw it in Southwest Missouri. And I didn't believe it. And so I sent some samples off to the University of Missouri, and they got on the phone to me. Very rarely, you send something into the plant diagnostic clinic, they just send you a report and the bill. But I actually got a call from the pathologist, and he said, "Where did you find this?" I told him, and a week later, he shows up on the, not on my door literally, but he called and he showed up, wanted to see it. This is in fact tar spot. This is not tar spot that I took pictures of, but you can see it does literally look like somebody just took a paintbrush and slung tar spot on there. Sometimes you will see it with like this fisheye around it. You can see that what we call necrotic tissue, it's just brown tissue around. Sometimes it doesn't have it. And you'll see it even when the corn starts to dry down, senesce. If you get common rust or southern rust, sometimes late in the season, the southern rust will form these blackish spots where the fungus is actually beginning to sporulate, and it tends to look like tar spot. And so I've gotten calls in the past saying, "Oh, I got tar spot in my corn." And when you would go look or send off samples, it was really just southern rust that they had missed. So yes, that is in fact tar spot. We're going to come back to that. Just keep that image in your mind. We're going to come back to that in that story here in a minute. Okay, how about this? Guessing you see this one a fair amount. I think you had it right the first time. That's gray leaf spot. The dead giveaway. Look at the leaf. Look how straight and how they stay within the veinal lines. They're very blocky. That is a dead giveaway that this is gray leaf spot. We say gray leaf spot. Sometimes it looks like brown leaf spot. But under certain conditions, it will tend to look like gray leaf spot. Another good way here, this is you want to impress your friends. You go out in the field and you find this, hold it up to the light and see how it's backlit here. And you see this yellowing, what we call chlorotic tissue around that. That's places where the disease is beginning to stretch out and it's starting to kill the tissue around it. And you can impress your friends. "Oh, I know what that is. We don't need to call the agronomist. That's gray leaf spot." So just a little trick that you can show them. And as that disease progresses, which it is highly infectious and it really likes to spread a lot if you get the right conditions, it can do a lot of damage and harm a lot of leaf surface, photosynthetic leaf surface. Now, if you've noticed, we've actually switched from resident diseases, those that are present in our soil, to migratory. In other words, this disease blew in on winds. Maybe it is a hurricane or something like that, but we've gotten that disease moving. Also, it's no longer cool, wet soils, but it's high temps and high humidities. That is relative. We're looking at above 80 degrees with above 50% humidity. That's kind of how pathologists describe high. And I tell them, come to Southwest Missouri. We'll show you what high temperatures are and high humidity is. So that's gray leaf spot. Don't confuse it. And I put this in here just in case we had some folks from Nebraska. But there is another disease that is over here in this picture, and you get sort of very similar lesions. It's called bacterial leaf streak. It'll look more yellow And if you get out around, well, Steve, where you are, Grand Island and that area, it really is beginning to take off and it can destroy a lot of crop. I was telling these guys, Reggie and Steve, Ken, I guess we were talking last night. That disease, I've seen it make zeros. Guys went to harvest and there was absolutely no corn out there whatsoever. It had taken that much tissue. Now, the problem, if you're really keen, you notice it's a bacteria. There's not a thing you can do once you have it as far as spraying a fungicide. It has to be controlled biologically through various means. And so just be aware of that. It is a soil-borne disease that you get and can be treated, just can't be treated utilizing normal methods. Okay, how about this one? You are correct. That is northern corn leaf blight. Northern corn leaf blight has these, everyone always describes them as cigar shaped. I don't smoke cigars. Haven't really been around a lot of cigars. When I think of cigars, I think of, what was that show? The Dukes of Hazzard. Remember the big fat boss hog? He smoked that great big old fat. That's what I think of. But they have a narrower end to them. They tend to cross the veins of the corn plant. Here it is backlit. And notice you don't see that bleeding out, that, you know, how we saw with gray leaf spot, that chlorotic sort of bleeding out into the tissue. But again, notice how damaging it can be to leaf tissue. Very, very aggressive disease that can take yield. So again, this is northern corn leaf blight. Why are we doing this? Well, the fact is, we can minimize our pathogen load by maintaining good, healthy plants. And that's why I go back to my story of walking out of that biologically controlled field where I wasn't looking for it, but I didn't see a lot of disease. And when we went into the conventionally controlled field, it was so obvious. You saw it quite well. And of course, that showed up even on Derek's tissue test. Remember when I told you about finding tar spot? Any ideas of what might be going on with these 3 ears of corn? Pretty big difference between them, isn't there? Any thoughts? You're exactly right. I was walking in this plot, a friend of mine that I used to work with, he said, hey, will you come and look at this plot with me and help me take some notes, that kind of thing. And he said, I've got some new hybrids and I'm not really sure how to evaluate them. And so we started walking and I found tar spot it started off in one particular hybrid, ended up finding it in multiple hybrids. And this was sort of right up front. I found very little tar spot on that particular, where this ear was taken. It maybe had a 10% incidence level. So if you took all the leaves off the plant and figured out there's about 10% of them that were covered by tar spot. This leaf had about 30% incidence level. Same row, about, I don't know, 1/8 of a mile down that road. Walked all the way to the other end. This one had about 40% incidence level on those leaves. Now, what do you think that yield differential would be? If you had that, one side of your field had ears like this, one side had ears like this, is that 15%? Yeah, it's a lot of yield. And you can see, remember the number that we talked about at the beginning, 15 to 30% yield loss. Well, when you go to harvest, you're going to be like, I never saw a problem with it. Can we increase or lessen our severity of disease infection by having good nutrition? The answer is yes. And the way that I like to think about this, if I hold my arms like this, My right arm is indicative of kind of corn yields. The yield is never higher than the day we open the bag. As a matter of fact, genetic corn potential is probably somewhere in the neighborhood of 700 to 800 bushels per acre. Can you imagine? I mean, if David Hula is making 600 bushel per acre and he's got yield suppressive effect, there's probably 700 bushel potential. And everything that happens throughout the growing season, pest, disease, poor nutrition, all of those things are reducing that yield. Soybeans are the exact opposite. The yield potential just continues to grow throughout the season. And so I think it's important to understand when we're talking about this, that the way in which we manage soybeans versus corn might be slightly different. The same with wheat, but it all comes back to this idea. We are trying to maintain an adequate nutrient balance in that plant to give it the best possible potential that it can have. And then, of course, I'm going to come back often to potassium and its role in disease mitigation, and then the role of manganese. With that, any questions about disease before I turn it back over to Gary? Questions? Any questions?
Yellow flash and brown early soybeans, which was generally a manganese deficiency as it was trying to
Yeah.
What the glyphosate out of the system, which caused manganese shortage, which then caused a disease problem.
Mm-hmm. Yeah, so oftentimes people, when we talk about herbicides, plants have to metabolize herbicides. It's not exactly true with glyphosate. Plants don't metabolize glyphosate. They sequester glyphosate and send it to the phloem. But guess what can't move when you've got your plant full of glyphosate? Manganese can't flow throughout, and so it begins to show up. And so it's what we call a sequestration deficiency. Essentially, it's showing up because right now the manganese cannot be sequestered. The other thing that happens, glyphosate goes down to a place in the plant right at the crown of a plant, and that's where the triggering mechanism for glyphosate resistant crops is that actually otherwise glyphosate resistant crops would die. And so it goes to this place and it looks kind of spiky like this. And what happens is normally glyphosate would fill one of these spots and that would starve the plant. But what happens is glyphosate resistant plants have an amino acid that builds up right here. And so the glyphosate sits on top and you're not able to actually take in that glyphosate. Guess where manganese likes to sit as well? Right here. And so that's why you get that yellow flashing effect. It's temporary, but it definitely is not a good thing that you want to be doing to your plants. And then we talked about the fact that glyphosate does break down, but it doesn't break down completely. It is still in the soil and bound to soil particles